So hi, my name is Audrey Brumback. I'm a pediatric neurologist and neuroscientist at the University of Texas at Austin. I am 80% research, 20% clinical. I have a neurodevelopmental child neurology clinic where I take care of kids with neurodevelopmental disorders, including autism. So the focus of my talk today is going to be on the thalamus and how thalamic physiology may play a role in disorders of the brain that are characterized by autism. So I have a great team at the University of Texas. Most of this work has been done by Polina Luboslavsky. So here we are in 2019, and I know that this conference is focused on a very specific genetic change, but in the overall, when a child comes into my office, they're not coming in with a specific genetic change, they're coming in with a set of symptoms and signs. And so what we've been able to do through syndrome, testing clinically, and then also through genetic testing, is understand how diverse the array of genetic changes are that can cause developmental encephalopathies. And so one challenge that we have in the field is how do we design treatments for these kids when everybody has their own different genetic etiology. So the focus of my work has been to understand how we get from this diverse array of genetic changes to common behavioral phenotypes. That's a picture of my daughters. And so the focus that I take is on circuits. So the idea being that if we can localize symptoms and signs to specific populations of neurons, specific circuits, then we can develop therapies that target those circuits that will treat the underlying, that will treat the symptoms regardless of the underlying etiology or genetic change. So my postdoctoral work focused on the prefrontal cortex, which has received a lot of attention in the autism field. Many autism-associated symptoms localized to the prefrontal cortex, including executive dysfunction, cognitive flexibility. So the prefrontal cortex has received a lot of attention, but the prefrontal cortex does not act in isolation. The prefrontal cortex was actually first defined by being the main downstream target of a part of the thalamus called the mediodorsal thalamus. And so these two brain regions are intimately connected. And what we're starting to understand is that in order for the prefrontal cortex to do its job, it actually needs inputs from the mediodorsal thalamus in order to do that. And that work has been mostly spearheaded by Mike Halassa at MIT. So in autism, functional and structural imaging studies have demonstrated that the connection between the prefrontal cortex and the thalamus is abnormal in people with autism. And so what I've done is, in my lab, is to focus on the thalamus and try to understand how its connection with the prefrontal cortex is influencing behavior and how that is abnormal in autism, and how targeting specific populations of neurons in the thalamus may be able to treat different symptoms and signs that are associated with autism. And so I'm showing here a mouse brain because I focus on mouse models of autism. And so how do we identify different populations of cells within a specific brain region? Well, one way is by the inputs and the outputs of those cells. So where do those cells project to? Who are they receiving inputs from? We can measure gene expression at baseline and then in response to activity. We can fill cells and measure their morphology, so their dendrite branching and their cell body configuration. And we can measure the electrical activity of these neurons and identify different classes of neurons based on any one of these circuit level phenotypes. So the there's been a lot of work done to characterize different electrophysiological neuron populations in the prefrontal cortex and other brain regions. And so that's where I started. Since I'm trained as an electrophysiologist, I wanted to understand if there were different electrophysiological circuits within the mediodorsal thalamus, this main input to the prefrontal cortex. So what we found in prefrontal cortex and in hippocampus is that long-range projection neurons, so neurons that are sending projections from prefrontal cortex to thalamus, from CA1 downstream to distant brain regions, they fall into these two categories, high sag and low sag. And so what does that mean? So I'm going to show today, the light blue will indicate high sag cells and the dark blue will indicate low sag cells. So in what we've seen is that in prefrontal cortex at least, there is selective vulnerability of high sag neurons. So what is this sag property that I'm talking about? Well, it's actually quite easy to measure. So I'm showing you here two neurons recorded in current clamp, and we're applying hyperpolarizing current steps. So what you see is that if you measure the difference between the voltage at the open circle versus the voltage at the closed circle, you have two different phenotypes. Some cells have a high sag and other cells have a low sag. And so we can talk later about, what causes that sag and what that might mean. But for the purposes of this talk, let's just use that as an indicator of what cell type we're referring to. So in prefrontal cortex, high sag neurons are selectively affected in autism models, whereas low sag neurons are not. So this is work from Dan Johnston and Darren Brager's labs, where they've looked in the Fragile X mouse model and observed that in prefrontal cortex, there's a very striking difference in the physiology of high sag neurons, but not low sag neurons. So as you see on the left, in the high sag cells, if you compare the black, which is wild type, to the red, which is knockout, you see a big difference just by eye in the physiology of these neurons. Whereas the low sag neurons, there's really not a big difference between the wild types and the knockouts. Okay, so a selective vulnerability in a specific population of neurons within the prefrontal cortex. And importantly, these are actually the cells that project to thalamus. So from the work that I've done, I also observed changes in these high sag neurons in the prefrontal cortex. And it wasn't just in one model of autism. I actually observed this in 3 different models of autism. And so this is where I start to think about circuit level phenotypes and if there are specific cell types that we could potentially be targeting for treatment because they are selectively vulnerable to the genetic changes. So here I'm showing you a control neuron on the top and a valproic acid treated animal on the bottom. Valproic acid is an anti-seizure medicine, and when pregnant women or mice are exposed to VPA midway through gestation, there's a high incidence of the offspring having autism spectrum disorder. So we observed that in 3 different mouse models of autism, there was a decrease in excitability in these high sag neurons in the prefrontal cortex. So what I'm showing you here is the action potential frequency, so just the number of upticks that you see in those traces on the left, counting the frequency of those as a function of how much current we're giving the cell. And so in each of these cases, we saw there was a decrease in excitability in just the high sag neurons of the prefrontal cortex, not in the low sag cells, which I'm not showing you here, but it's published. Okay, so long-range projection neurons segregate into these 2 populations, high sag and low sag. And in prefrontal cortex, high sag neurons are selectively affected in autism models. I'm not going to show it to you today, but this work is published. There's been years of research examining CA1 cells, so hippocampal neurons, the main output cells of the hippocampus, and how they also divide into this high Sag and low Sag categorization. And those cells not only differ in their electrophysiology, but also in their morphology and their connectivity patterns. And so the question I have is, what about the thalamus? So what we're starting to understand is that the prefrontal cortex does not act in isolation. It really needs the thalamus in order to do its job. And so what I want to understand is, are there populations of neurons in the thalamus that are also selectively vulnerable to differences in genetics that are associated with autism? And could those cell populations become circuit-level targets for treatments for these prefrontal associated symptoms? So I have two hypotheses. The first is that the neurons in the mediodorsal thalamus segregate into these high Sag and low Sag classes. And two, that in the Fragile X model, the high Sag neurons will be selectively affected as they are in the prefrontal cortex and in CA1. So in order to isolate the specific neurons that project from thalamus to prefrontal cortex, we inject a retrograde tracer into prefrontal cortex that gets trafficked back to the cell bodies in the thalamus, and we can record from glowing cells in the mediodorsal thalamus. And so, as you can see, each one of these bright spots is this individual neuron that projects from thalamus to prefrontal cortex. So, to address hypothesis one, are there two populations of neurons that divide by this Sag property? There are. So, when we record from a large number of neurons in thalamus, we do see that they segregate very nicely into these two populations. You can see on the right, when we look at the voltage-Sag ratio, there's clearly a bimodal distribution. So when we look at the properties of these cells, it's not just the Sag that's different. They also have different subthreshold electrophysiological properties. So on the left, I'm showing you that voltage-Sag ratio, and then in the middle, I'm showing you the resistance of the cell, which is a contributor to cellular excitability, and then membrane time constant, which affects how well cells can integrate different inputs. So what we see is that there's clear differences between the high Sag and the low Sag neurons in wild-type mice. And when we examine these properties in Fragile X knockout mice, we see that those properties are really not different. So we don't see a difference in the voltage-Sag ratio, in the resistance of the neurons, or in the membrane time constant. However, these cells have a really prominent defect in their ability to generate action potentials. So what I'm showing you on the top is high Sag neurons in wild type versus high Sag neurons in knockout mice. And on the bottom, I've quantified that action potential frequency as a function of the amount of current we're giving the cell. And you can clearly see that there's a large difference between the wild type and the knockouts. The knockouts generate fewer action potentials. And we do not see this in low Sag cells. So this is a selective deficit just in these high Sag neurons. So the thalamic neurons have a couple of different ways of firing action potentials. They can fire bursts of action potentials or sort of slow, steady trains of action potentials, so burst or tonic modes of firing. And what we're seeing is that these high Sag neurons have defects in both types of firing. They have defects in the tonic firing, which is what I just showed you. And then when we measure just the number of action potentials in bursts, we also see a defect that is also not present in the low Sag cells. So what I've shown you is that cells that project from the thalamus to the prefrontal cortex segregate into high Sag and low Sag populations, just like they do in the prefrontal cortex and hippocampus. And loss of the Fragile X gene causes hypoexcitability in just the high Sag cells. So next steps are really to understand, do these high Sag and low Sag cells belong to different circuits? And so what we're doing is we're characterizing these neurons in terms of their morphology, their afferents, their efferents, their gene expression, and their morphology in order to understand if these are in fact distinct anatomical populations of cells that could potentially be targeted for therapy. And then long term, the question is really, is this selective hypoexcitability of the high Sag neurons, is that a common circuit level phenotype as it is in the prefrontal cortex? We saw in the prefrontal cortex that multiple animal models of autism shared defects in high Sag neurons. I predict that we are going to see a similar circuit level phenotype in the thalamus when we examine different underlying etiologies of autism. And so this is all with the overall goal of trying to localize symptoms and signs to specific circuits to develop circuit level therapies to treat symptoms and signs regardless of the genetic cause. Thank you.